Hello students. In my previous class, we have studied about the culture methods of shrimps. There we have studied that is the extensive method, semi-intensive method, intensive method, pond culture, polyculture. Now, in today's class, we will discuss the remaining three types of cultures, that is the culture in berries, pen culture, cage culture. Okay, culture in berries, pen culture and cage culture. Now, what is the culture in berries? Okay, berry is a low-lying area or a land, okay, near the coastal area, filled with the brackish water, Culturing the shrimps in these berries is called as culturing berries. It is a type of extensive method. Okay, it is a type of extensive method. Here in this tank, that is during the month of January and February, when there is no water in this area, it is embanked. Okay, it is what? Embanked. The side, a marginal area is what? It is embanked, and then the water is stored. In this type of culture, the supplementary food is not provided. Okay, the supplementary food is not provided. Whatever the natural feed is there, on that feed only this berry culture is carried out. So, what are the sequences in this berry culture? I told that berries are lowland situated near the estuaries. Okay, lowland areas near the estuaries, and then during the month of January, there is no water in these tanks. Okay, there is no water in these tanks, and these tanks are then embanked. Okay, then these are embanked and made around that the tanks. Okay, now during the high tides, okay, that is usually for the full moon and new moons phases, there will be high tides, and these high tides are brings the shrimp seeds into the berry tanks. Okay, they bring the shrimp seeds into the berry tank, and this, along with these shrimp seeds, the nutrient materials also enter into the berries. Is it clear? So, the entry of the water is carried out through the sluice gates. Okay, there are what? Sluice gates, and through these sluice gates, the water enters into the low-lying areas. Okay, low-lying areas which are nothing but the berries. Now, the sluice gates are connected at the connecting points of the berries and feeder canals. Okay, feeder canals. Now, this seawater is usually introduced in the month of February, March. Okay, February, March. I told in the month of February, March, at the time of full moon and new moon periods, there will be high tides. And these high tides brings the shrimp seeds into the this berry tanks. Okay, this into the berries, and then these are cultured. And up to the month of October. Okay, up to the month of October, these will grow up into the marketable sizes, that is 7 to 12 centimeters in length. And once they are marketable size, when they grow to the marketable size, they are harvested. The shrimps are caught and sent to the market for sale. The production of the shrimp is seen as 400 to 600 kgs per hectare. Okay, per hectare. So, in the berries here, that is, this is the one C is there, that is, then this C, you will see that the estuary, at the estuaries, there is a sluice gate. Okay, there is a sluice gate. Now, this sluice gate helps in entering the water, and as well as it can be controlled, the exit of the water is completely controlled with the help of this sluice gate. Now, along with these sluice gates, okay, the water enters during the high tide, the brackish water enters into the berry, which is a low-lying area. Then once it enters, it is usually entered during the high tides in the month of February, March, new moon and full moon phases. Okay, so that water is now stored and stopped with the help of the sluice gate. They are reared in this tank. Okay, and up to the October, by the month of October, they grow to the size of 7 to 12 centimeters, and once they grow to the marketable size, they are harvested Okay, they are harvested by opening the sluice gate, etc. Okay, so that is about the culture in berries. Okay, now the another one, that is the pen culture. Okay, pen culture. Now pen culture is also it is an enclosure made in a water. Okay, it is an enclosure made in a water by the nylon netting, that is, for example, the one side, two side or the three sides of are made with the nets, covered with the nets, and the one side is left with the natural bank. Okay, that is one or two or three sides. The fence is constructed. See here, that is, the fence is constructed from one side, two side or three side, and one side is kept with the natural bank. Okay, so that there should be an entry of the water. Now this appears as if a duck fence on a land. Okay, this pen culture, if you see it, it appears or resembles like a duck fence on the land. Now the pen may be, okay, this pen may be single layered or double layered. Okay, single layered or double layered. Usually single layered is used for the adults. Okay, single layers are used for the adult shrimps, whereas the double layered is used for the larvae. Okay, double layered is used for larvae. Now how it is there? There is a split bamboos. Okay, split bamboos which are pointed at one end. Okay, which are pointed at one end. They are placed or put into the soil or the mud. Okay, into the mud, and then it is covered with the nylon net. Is it clear? Then that these woods are usually what? Of the casuarina pole. We call it is what? Casuarina pole. Now these, and then the pole should be kept, that is the 0.5 meter. Okay, you can see here the 0.5 meter of these poles should be left above the water level. Okay, they should be kept above the water level. Then the mats are supported by the casuarina poles. Now in this pen, the density of the shrimps should be 10 to 100 prawns or shrimps per square meter. Okay, density should be what? 10 to 100 prawns per square meter, and the yield is 6 tons per hectare per year. Okay, 6 tons per hectare per year. So what is this pen culture? Pen is an enclosure in a water made up of the nylon nets. Okay, pen is an enclosure in a water made up of nylon net. Okay, for the shell fishing. Now this pen resembles, I told, the pen resembles like a duck fence on a land. How it is covered? It is covered from one side, two side or three side. Okay, leaving one natural bank. Okay, at one side there should be a natural bank. Then the pens may be single layered or double layer. Single layer pen or the nets are used for the adults one, whereas the double layered is used for the larvae. Okay, then the split bamboo measuring about 2.5 meter height, okay, with the pointed tips are nailed horizontally, placed in the reapers. Is it clear? These are, okay, so they are placed in the water, leaving that on the tip of this, that is the 0.5 meter of the top portion of these bamboo poles should be kept above the water. Then the mats are the supporting for these casuarina poles. So what should be the stocking here? The stocking of these prawns or the shrimps should be 10 to 100 for every square meter. In the pen culture, the yield is about 6 tons per hectare per year. So the berry culture, pen culture, the another one is the cage culture. Now the cages are the suspended boxes in the water. Okay, cages are the suspended boxes in the water. It's the size of the cages depend upon the size, the strength of the culture. Okay, that's what the strength of the culture or the density of the culture. Now the square or the rectangular in shape. Okay, they may be what? Square or rectangular in shape. What is done here? So this is what? The cages. Okay, the cage is made up with the iron frames. Okay, iron frame covered with the nylon net. It should have a lid at the top. Okay, it should have the lid at the top. This is the one of the cage. Is it clear? So the frame is what? The iron frame. Then it should have the lid. and it is the entire thing is covered by the nylon net. In this cage there should be sinkers, okay, there should be sinkers at the base, okay. To the cage we will be attaching a sinkers at the base and floaters at the top, okay, so that the cage is suspended, so that the cage is what? Suspended in the water. The cage is not allowed to touch the bottom or the mud. Is it clear? So that's what here, if there is a sinkers at the base and floaters at the top, it will balance the cage in the water without touching the bottom of that water body. Is it clear? So that is the cage. If the cage is in a square shape, it should be of 1 into 1 into 1 into 1 meter size, okay, length into breadth into height into depth, okay, size, and is enclosed on all sides with the polythene monofilament webbing. Polythene monofilament webbing of 2 millimeter mesh size. Is it clear? So that is what here I told the stocking density in the cage should be what? 10 to 20 numbers per square meter. The density in the cage should be 10 to 20 numbers for every square meter. That is the cage culture. So cage is what? Nothing but a suspended boxes in the water made up of nylon netting. Okay, then these cages, the frame of the cage is made up of iron. It should be covered with the nylon nets. That is what? The polythene monofilament webbing of 2 millimeter mesh size. Now, the cage should be having a sinkers at the bottom and floaters at the top, so that the cages should be suspended in the water and should not get settled at the bottom, okay, should not get settled at the bottom. So that is what here in the cage, the density of the shrimps or the prawns is 10 to 20, okay, 10 to 20 in numbers for every square meter. So that is what the culture in Bheri, then the pen culture and cage culture. With this we have discussed the different culture methods, that is the extensive method, semi-intensive method, intensive method, pond culture, Pockley culture, then the culture in Bheri, then the pen culture and cage culture. With this we have finished the different methods of culture of shrimps, okay, different methods of culture of shrimps. So in my next class we will discuss about the freshwater prawn cultures, okay, freshwater prawn culture. There also we will study the different methods of seed collections, then we will study the different methods, cultural operations, etc. Okay. So thank you.